Hey there, health enthusiasts! Welcome to our latest roundup of fascinating medical breakthroughs and health discoveries that are making waves in the scientific community. Today, we're diving into everything from cancer research to anti-aging science and brain health. So grab a cup of tea, get comfortable, and let's explore these exciting developments together. Stanford medicine researchers have made a groundbreaking discovery in cancer treatment by developing a method to reactivate cancer's molecular kill switch. This approach harnesses our body's natural process of apoptosis, the programmed cell death that eliminates billions of cells daily. Dr. Gerald Crabtree's team created a molecular glue that artificially brings together two proteins, BCL6, which when mutated drives blood cancer, and CDK9, which activates gene expression. When these proteins interact, they trigger cell death genes in cancer cells. What makes this approach revolutionary is its specificity. Unlike traditional chemotherapy that kills healthy cells alongside cancerous ones, this method targets only the problematic cells. As Dr. Nathaniel Gray explains, you take something that the cancer is addicted to for its survival and flip the script, making that very thing kill it. Scientists from Osaka University have created an AI model that can estimate biological age from just 5 drops of blood. Unlike chronological age, biological age measures how well your body has aged at the cellular level. The AI analyzes 22 key steroid hormones that play crucial roles in metabolism, immune function, and stress response. What makes this approach clever is that instead of looking at absolute steroid levels, which vary widely between individuals, the model examines the ratios between different steroids. This reduces the noise caused by individual differences and focuses on meaningful aging patterns. Our bodies rely on hormones to maintain homeostasis, so we thought, why not use these as key indicators of aging, explains Dr. Qiuyi Wang. This tool could offer valuable insights for personalized health management and help identify people at higher risk for age-related diseases before symptoms appear. As we age, our bodies accumulate zombie-like cells, senescent cells that no longer divide but refuse to die. These cellular zombies cause problems by secreting inflammatory molecules, contributing to chronic inflammation throughout the body. This inflammaging has been linked to numerous age-related diseases. Researchers at Sanford Burnham Prebys have discovered that mitochondria, our cells' power plants, control the ability of a DNA repair protein to suppress this inflammatory program, known as the senescence-associated secretory phenotype. In addition to no longer growing, the hallmark of senescent cells is this inflammatory program causing them to secrete inflammatory molecules, explained Dr. Peter Adams, senior author of the study. The research suggests that targeting mitochondrial pathways could potentially reduce the chronic inflammation that accompanies aging, opening up exciting possibilities for developing interventions that might slow aging and reduce the risk of age-related diseases. CDKL5 deficiency disorder is a rare but devastating brain condition affecting young children, characterized by early-onset seizures and severe neurodevelopmental impairment. The disorder is caused by pathogenic variants in the cyclin-dependent kinase-like 5 gene, which regulates key phosphorylation events essential for proper brain development. Though relatively rare, affecting up to 1 in 40,000 live births, CDKL5 variants are among the most common genetic causes of severe childhood epilepsy. Initially considered a variant of Rett syndrome, CDD is now recognized as an independent disorder with distinctive characteristics. What makes this condition particularly challenging is that the pathogenic variants either result in complete loss of CDKL5 catalytic activity or led to partial loss of function. Researchers are making progress in understanding how CDKL5 functions in human neurons, including identifying phosphorylation targets. This expanding knowledge offers hope for targeted drug discovery that could significantly alter the disease trajectory in young children. Our understanding of memory formation continues to advance through innovative research techniques like optogenetics, a powerful tool that combines genetic and optical methods to manipulate specific brain regions with unprecedented precision. Recent studies have revealed fascinating insights into how different brain areas interact to control memory processes. By using light to activate specific neurons, researchers can observe the resulting activity patterns in real time. One discovery involves the ventrolateral orbitofrontal cortex, VLO, which appears to have profound global influence on brain activity. When scientists stimulated excitatory thalamic projections to the VLO, they observed opposing effects depending on the stimulation frequency. Low-frequency stimulations led to widespread decreases in brain activity, while high-frequency stimulations caused widespread increases. Similarly, studies involving the hippocampus showed that activating this memory center produced significant responses not just locally but in distant regions, including the frontal lobe and striatum, demonstrating how memory processes involve complex networks spanning multiple brain regions. Researchers at UConn School of Medicine have uncovered a critical clue that might help explain the cellular mechanisms behind devastating neurodegenerative diseases. Their study reveals that decreased levels of the TDP43 gene may be significantly contributing to conditions like Alzheimer's disease, frontotemporal degeneration, and amyotrophic lateral sclerosis, ALS. Lead author Omar Mustafa Fathi conducted this research in Dr. Patrick Murphy's laboratory, collaborating with Dr. Rikyang Yan, a leading expert in Alzheimer's research. While clinical evidence has long suggested that blood-brain barrier dysfunction plays a role in neurodegeneration, this study specifically explores the contribution of endothelial cells, the cells that line blood vessels and form this critical barrier. The findings provide a novel perspective on how vascular problems might contribute to these brain diseases, exemplifying the powerful collaboration between vascular biology and neuroscience research. This work could potentially open new avenues for treatments that target the blood-brain barrier, addressing underlying causes rather than just managing symptoms. A fascinating study examining the connection between socioeconomic circumstances and biological aging has revealed that children from disadvantaged backgrounds may be aging faster at the cellular level. Researchers analyzed saliva DNA methylation from 600 children and adolescents participating in the Texas Twin Project using a novel measure of aging called Dunedin-POAM. The results were striking. Participants living in more disadvantaged families and neighborhoods exhibited a faster pace of biological aging. Additionally, Latinx-identifying children showed accelerated aging compared to both white and Latinx white-identifying children, consistent with the higher levels of disadvantage often experienced by these groups. This measure wasn't correlated with chronological age in this young sample, suggesting it's capturing something distinct from normal development. These findings lend scientific weight to the observation that chronic stress can get under the skin and affect health at the cellular level, potentially explaining why socioeconomic disadvantage often translates to higher disease risk later in life. Scientists have developed a new CRISPR-based test that can detect pathogens directly in blood samples without requiring DNA amplification steps. This breakthrough could revolutionize how we diagnose infections, particularly in resource-limited settings. Traditional diagnostic tests often require complex equipment to amplify genetic material, making them difficult to deploy in the field. This new approach leverages CRISPR technology's precise targeting abilities to identify specific pathogen DNA sequences, even when present in very small amounts. By eliminating the need for amplification, the test is faster, more portable, and potentially more affordable. Early validation studies show promising results for detecting bacterial infections like sepsis and viral infections including HIV and hepatitis. The system uses a specially designed CRISPR protein that, when it finds its target sequence, activates a fluorescent reporter molecule detectable with relatively simple equipment. This technology could dramatically reduce the time from sample collection to diagnosis from days to just hours, potentially saving lives by enabling faster treatment decisions. How much sleep do you really need? According to the latest research, there's no one-size-fits-all answer. Your optimal sleep duration depends on multiple factors, including age, genetics, activity level, and overall health. While the general guideline for adults is 7 to 9 hours per night, some people genuinely function well on 6 hours, while others need a full 9 to feel their best. What's becoming increasingly clear is that sleep quality matters as much as quantity. Deep sleep and REM sleep phases are crucial for cognitive function, emotional regulation, and physical recovery. Recent studies using wearable technology have revealed interesting patterns. Many people who thought they needed less sleep were actually experiencing micronaps during the day or suffering cognitive deficits without realizing it. The research also suggests that consistency may be more important than total hours. Maintaining a regular sleep schedule helps optimize your body's circadian rhythms, potentially offering more benefit than occasionally catching up with a long weekend sleep. Tuberculosis, one of humanity's oldest diseases, has remained a persistent global health challenge, until now. Recent breakthroughs are transforming our approach to this ancient disease that still infects over 10 million people annually. For the first time in over 50 years, we're seeing the development of new TB medications that can significantly shorten treatment duration. Traditional TB treatment required taking multiple antibiotics for 6 to 24 months, making adherence extremely difficult. New regimens can potentially cure some forms of TB in as little as 4 months. Additionally, rapid molecular diagnostic tests now allow for diagnosis in hours rather than weeks, enabling faster treatment initiation. Perhaps most exciting is the progress in vaccine development. Several candidate vaccines are in late-stage clinical trials that could provide better protection than the century-old BCG vaccine. These advances, combined with increased funding for TB programs, are creating unprecedented momentum in the fight against this disease, with personalized treatment approaches potentially transforming outcomes for millions of people worldwide. A comprehensive study has found no link between COVID-19 vaccination and major structural birth defects. This large-scale research examined pregnancy outcomes in vaccinated versus unvaccinated women, following them from early pregnancy through birth and early infancy. The findings showed no increased risk of major congenital anomalies in babies born to mothers who received COVID-19 vaccines during pregnancy. This research is particularly significant because pregnant women were excluded from the initial COVID-19 vaccine trials, leading to understandable concerns about vaccination during pregnancy. The study tracked various birth outcomes including heart defects, neural tube defects, limb abnormalities, and other structural anomalies, finding no statistical differences between the vaccinated and unvaccinated groups. Public health officials are highlighting these results to reassure pregnant individuals that COVID-19 vaccination is not only safe, but beneficial, as it protects both mother and baby from potential severe COVID-19 infection, which itself poses significant risks during pregnancy. That wraps up our health news update for today. From revolutionary cancer treatments to insights on aging and sleep, these advances demonstrate how rapidly medical science continues to evolve. What's particularly exciting is seeing how researchers are finding new approaches to old problems, whether that's harnessing natural cell death to fight cancer or using AI to understand the aging process. Remember that staying informed about these developments is an important part of taking control of your own health journey. Thanks for joining me today. If you found this information valuable, please hit that like button and subscribe for more health updates. Until next time, stay curious and take care of yourself.